everybody's busy, so I'll be quick and short, but most of people may have already heard about FDA, but not many people heard about National Center for Toxicological Research. We are part of Food and Drug Administration's, only we are away from the Washington, D.C. with all the hustle and bustle and all these things we heard on the TV and all the time. Our main goal is to do the basic research in the area of toxicology, and as you can see, we are located in 35 miles south of Little Rock, and I want to invite anybody who wants to come, welcome. I'll be happy to show arms. I think Byron has been there one time, so I asked Anna and Alex, you're welcome to come and see the facility, and definitely, open the opportunity for collaboration between UT as well as, and I have an appointment at UT for almost 20 plus years, but it was all, the person I was in touch with already retired. So I'm glad to start a new collaboration with Anna, and I'm looking forward to more visits, and hopefully you guys can, I work for government, so this is definitely always a disclaimer because you don't know who is in the audience, that is, I don't know, everything is what I'm saying, not reported by or endorsed by the FDA. As Anna mentioned briefly about the International Drug Users Society, before I start my talk, I just want to give you a few slides. As a society, we started about almost now 10 years old society, and the main goal was the society to promote, collaboration between scientists. And this is really, the seed came from, I used to go to CPDD or other society, like SNERD meetings or INRC, and so we said, well, why not everybody can work together, and maybe we can bring people together. And as I will show you, I work on a neurotransmitter systems, and most of the drugs affecting through this, so I said, let's try and have a society where we can bring scientists together and they can have more collaboration, interactions, and understanding about each other's. And I would like to really invite who's working in the area of addictions that please join the society. It's one of the cheapest society to join, and I think it's very interesting, very good people who are part of the society. And the best thing is that we have always meeting in a very nice place. That's our president, current president is George Coop, and we had a last dinner at the Neuroscience Society, and we were talking about having a next meeting. We said, I like to have a nice place, the weather is good, food is good, and scientists definitely should be good. So as you can see, we had a meeting in Mexico, South Korea, Turkey. Last meeting was in Australia, Sydney, and we're going to have next month in Dubrovnik. And I met Malik that it's one of the really beautiful places. So I definitely would like to invite people to please join me. And I'll share you some of the pictures. This was in Istanbul, Turkey, Mexico City, Sydney, Australia, and this is Dubrovnik, so you can see. So that could be a good reason to go to the meeting. So I definitely would encourage all of you to join the society, and we're going to have a meeting, and hopefully it will be a good meeting. We also run a journal. I run a journal called Drugs and Alcohol Research. This is published by Aston Publishers. This is 3 years old journals, and we also try to put special issues. We put in a special issue on advances in neurobiological basis of inhalants. This is one of the symposiums we organized in Mexico, and to promote our journal, and these are the co-editors for that issue. And once we publish this, to promote this, we translate into several different languages. So this we're really kind of promoting around the world. We have a special, the introduction in Chinese, French, Japanese, and Portuguese, Russian, as well as Spanish. So that's really kind of give you more breadth of the journals of what we do. Anna just published a nice paper in the journal. I just want to show it, and I want to welcome any other people who have a paper sitting on the desk. Please submit the journal. We try to get a fast review, and we publish. This is an online journal available to anybody anytime. So it's really, as soon as we accept it, it's published. So it won't take more than a few days after this. So I definitely would like to encourage people, who may be looking or sitting on, wants to publish. Just a few slides about NCTR, National Center for Toxicological Research. As I mentioned, we conduct the peer-reviewed research. We provide the data for the regulations. We develop the new methods, techniques, procedures, and we design the goal is to better risk assessment for the new evaluated drugs or food contaminants or the devices and all these things. And this is kind of give you the chart where we stand. Like you have a HHS, we are part of HHS, like NIH, CDC, FDA, and within the FDA we have several centers, and we are part of that. And as you look at the scheme of this, just like a department, we have an office of directors, and within the directors we have an office of research, different divisions. As you can see, we are doing work in cancer research, personalized medicine, system toxicology, and I work in this division, neurotoxicology, plus we have other support for it. As far as the research is concerned, every time we start a project, we really want to do a very thorough research. So this is kind of like our research approach when we start a compounds or drugs or devices. We look at the if you look at the neurochemical or neurotox parts, we look at the neuropathology, neurochemistry, neurophysiology, as well as the behaviors, and what we call the neurotoxicity profile of any specific compound. We want to make sure, because sometimes it is, you may see some effects in neurochemistry but don't see the behavior, or you see the behavior but not electrophysiology, or you see the changes in behavior but nothing inside. So we try to understand in drugs or in a really complete way how it's working and what is the mechanism of action. We always try to correlate with the structural neurochemical lesions with the neurophysiology as well as behavior manifestation. And of course, we always look for the pharmacokinetics and differences between the species, because we have a nice facility for mouse models, rat models, rabbit models, primate colonies. So we try to exploit all these things. And this is just to show you the breadth over the 33 years I have done. I have worked with all different drugs of abuse, you name it, and I have worked with not only drugs of abuse, we have also worked for because we work for FDA, there's always sometimes questions, so this could be a good drug for treatment for alcohol, treatment for cocaine, treatment for, and as you can see, we have worked with ibogaine, fenfluramine, ayahuasca, ketamine. This is a very interesting alkaloids. They still use in Brazil for ritual, as well as they use for hunting in Africa. And there was a person in New York. He passed away now, but he was a heroin addict. So he took this compounds, ibogaine, and he really, from there on, he stopped taking the heroin. So he was very entrepreneurial. I guess he was from New York. He said, okay, he's going to make money. So he, just because of that, he tried to patent these compounds for the heroin, for alcohol, for nicotine, for all these things. But there was really no research was done. So he was start using political arm or went to the senator, to the congressman, and said, listen, FDA is not doing their job. We need to get this approved so you can help people can stop with taking drugs and everything. We have no clue what was that. So he, the senator from New York, called the FDA commissioner. So why are you not approving these drugs? And so when you look at the literature, there was nothing on the drugs. There's a procedure. You have to look at the, just to see how it affecting, how it, protecting the effects, how it's stopping the addiction. Does this has any adverse effects? Does it has any abuse liability? We need to do it. Also, you need to do it. That is so this is what it dumped on us actually. That we got this news that said, okay, we got to work on ibogaine. We need to find out if ibogaine has any adverse effects. It has an abuse liability. It has a neurochemical effect. How it is working? This person took it and never took the heroin. So we started working on these compounds, and believe it or not, this compound, we published almost 20 papers on that now. that this was more toxic than any of those drugs combined. Literally, we have published papers showing that this was produced in cerebral vascularizations. I mean, this is amazing, you know. And so really, when we published, he was still alive. He hated our guts. You know. These people killed him. And what he did, he also have a metabolize of this. You say, well, metabolite would work, you know, but it never went anywhere. But just an example, you know. Recently, we are getting another same crazy thing, say, well, ketamine is a drug used, as they use in anesthesia. There's several papers coming up, it's the treatment for alcohol and in depressions. And we just have couple of meetings last several days. We want to see if it has any adverse effects. And we are going to do pretty, in-depth study trying to understand that what is really happening. So something like this is really sometimes things projects fall on our back that we have to do it. Nobody wants to do it. We also have done a lot of work on food contaminants, some of the metals, therapeutic, agents, and we always use this as a model for Parkinson's disease. However, my love to the brain is a dopamine. I'm sure if you take a course in pharmacology, you have to know this compound, this molecules in the brain. This is a well-known neurotransmitter. It was discovered by Dr. Alvin Carlson in 1950. Actually, it was and I have a pleasure, actually, I've been working for dopamine for 1975. That's the first time I measured the dopamine in the brain by spectrophotometer, I still remember. And after all these years, I have a colleagues in Japan, he had a special symposium, so he invited Arvind Carlson, and I happen to know him, Toshi Narahashi, so we chaired the same session. I was so pleased, I was so thrilled to meet somebody who discovered the dopamine, and he really talked a lot about that. It took him 10 years to convince the scientific community that there's a dopamine in the brain. He has stories after story that at the time people were thinking he's a crazy man, he's a crazy young postdoc, and he's just nuts about this dopamine. And when he presented over and over, he was presenting in the Physiological Society at the time. Brady was there, and other people from England and Germany. Everybody told him, no, you're nuts. There is no dopamine in the brain. And he was persistent. He said it took him constantly, he did everything he could to justify there's a molecule in the brain called dopamine. So finally, in 1950, actually, they accepted that. This was a picture in Japan, actually, we were talking about. And I'm sure you know that structure. This is the circuits of the dopamine in the brain. So you have a cell bodies, and it's really good everywhere if you look at it carefully. And there's a lot of addiction work being done on this circuit of the brain. And the reason I am very, I love this because all the addiction drugs, any you can see not only the drugs but food, all these things one way or other related to dopamine. I look at all the literatures. As I say, I was so crazy that I have a song about dopamine. I have a musician, and I have a friend, a musician, he writes songs. So I provided him all the data. If you have a few minutes, I'll show you. I have the song at the end. But I provided him all the data about the dopamine, what he does. He has no clue. He's not a scientist. He writes songs. So he tried to manage, and it's pretty interesting. It's a work in progress. We are going, so I'll let you enjoy that song. So anyway, just to see how dopamine affects in different process. You can see what drugs. It affects methamphetamine, it affects the nicotine, it affects the PCP, it affects I have the 3X, which I call sugar, salt, sex, and fat. It affects, actually, if you look at the literature, there are effects of these things on the dopamine. And literally the whole page is full of all these compounds, one way or the other, affecting the dopamine system. And as I mentioned in the very beginning, I have colleagues who work on the cannabinoids. He really disputes this. The dopamine has nothing to do with addiction. So we always argue about that. You can see caffeine, very simple, sugars and everything. So that just show you that how many drugs, or even the people have reported, actually there was a meeting, it was in Italy, showing is the shopping, Literally they have the MRI in the brains, and you can see people were flashing the cards and all different types of much net and dopamine peaks. Anyway, so far it's just an introduction. Let me talk about what we are doing and what we have done almost last 10 years about the methamphetamines and effects of dopamine and how nitric oxides play a role in the methamphetamine-induced dopaminergic toxicity. As you can see, this is a meth lab in North Carolina. It's very easy. This is a good thing about meth. You can make from anywhere. This was the back of the countryside room. They found it, and right there you can make all the meth is possible. The thing about methamphetamine is a very widely abused legal drug. It can manufacture, distributed, abused around the world. And not only us is the problem in Asia, problem in Europe, definitely North America, and it can be injected, snorted, smoked, ingested. It goes so fast to the brain that is really amazing. Okay? It doesn't matter. You don't have to be only like with heroines and all these things. You have to inject it. This is you can inhale it and it's there. And you can see some of those surveys showing that as you can see, the abuse is going higher and higher. This is a political map, and you heard about the blue state versus red states. You can see, I think yours is in a blue state, Tennessee, but we are red. We are really pretty high, Arkansas is pretty bad. This is my favorite chart. What it is, this was a survey done by the, how many meth labs was confiscated in each state. And you can see Arkansas is at 543. And if you know, the Arkansas population is only 2.5 million. I was comparing with California, it's 25 million people. Can you imagine the ratio? It's amazing. And the reason is that half of Arkansas is really all the natural state and has the national park. So as I showed you before, if you have the back of the truck, you can go there, nobody's there, you can synthesize this and sell it. So really, it's amazing how much going on. This is again showing the survey from almost 20 years that meth abuse is going higher and higher. It also shows that different surveys shows it goes up. And this is also interesting to see that 23.4 billion in use in the treatment for drug abuse and addiction, especially in that. Like I say, it's not only problem in US. I was in Japan and they found that they said that North Korea, they discovered a whole ship and they were a factory which was using meth, synthesizing the meth and shipping to the Japan. They confiscated a whole ship, loaded with it. In Mexico, I was there. They confiscated $200 million cash from the Russian drug cartel. Can you imagine looking that kind of cash? Literally get it. Anyway, so what we did, we took some C57 mice, we gave some methamphetamine, 10 milligram per kg body weight 4 times. And we took the strata, which is part of the brains, and we looked at the dopamine concentration. This is control. This is after 24 hours, 72 hours, 1 week, up to 2 weeks. And we have done up to 1 year. It's never back to normal. However, we also noticed that when these animals, when we giving the methamphetamine, the temperature was going up. So we took the animals, and actually that was happened by accident, really, because I was talking, we were doing this work, and I said, well, my technician, these animals are really getting hypothermia now. Why don't we go put in a cold room, actually. At that time, it was a lot easier than now. But we went and put the animal in the cold room, and really, when we took this animal, surprisingly, the dopamine was restored. This was controlled, 4 times 10 methamphetamine at room temperature. We took the 4 times 10 meth and put in the cold room. When I saw that, we said, well, let's increase the dose. And well, literally, this is the toxic dose. And they were able to, it's still alive. and their dopamine was not as depleted as after 4 milligram at room temperature. So I said, well, this is definitely, temperature is playing a major role. So what we did a couple of pharmacological compounds. Yeah, I'm sure you know phenobarbital, diazepam, MK801. They all reduce the body temperature. So we treated the animals, and we again look at the dopamine concentration. As you can see, control after 24 hours. This is after phenobarbital, diazepam, and MK801. They're all showing in some protective effects. So definitely, if you can block the temperature, you can restore the effects. So over the years, we have published a series of papers showing that it definitely affects the concentration of dopamine and serotonin. It decreases the activity of the key enzymes. It decreases the number of uptake sites, and a lot of histological evaluations show the dopamine degenerations. However, the mechanism is still not clear what is really happening. So we hypothesized that I think this is producing the oxidative stress, it's producing excitotoxicity as well as the energy loss. And this is all combining together, producing the neuronal damage. And I'll show you one at a time. This definitely, indeed, that was the case. So what happened when you give the methamphetamine, you have a flood of dopamine, you have an excessive release of dopamine, and because of that, you produce the autooxidations. And because of autooxidation, you're generating the free radicals. You are generating superoxide radicals. You're generating the hydrogen peroxide. With the phantom reaction, you're producing the hydroxyl radicals, and increase the lipid peroxidation, produce cell death. And if you have some of the antioxidant enzyme, which is we have always in the system, which can detoxify this. So if we have superoxide dismutase, it can detoxify to H2O2. Otherwise, it will show that toxicity will increase the ROS formation. So what we did first experiments, we took the transgenic animals with the strength for 2183. This is the higher level of copper zinc SOD in these mice. And we transgenically made those animals and did the very simple experiments. We took the animals, we look at the non-transgenic animals, injected methamphetamine. You can see the same replications, whereas SOD transgenic animals show a significant protection. So really, if you can increase the antioxidant activity by increasing the superoxide dismutase, you can detoxify that effect. However, so what happened after the next steps? If you have a superoxide and you are having nitric oxide available, you're generating a very short but very powerful peroxynitrite. And that may be able to produce the cell death. However, when we look at what kind of forms we are really looking, nitric oxide, when you look at the body, you got the eNOS, iNOS, and nNOS. eNOS is mostly smooth muscles, iNOS is the microglia, as well as the macrophages, and nNOS is what we are interested in. So you have nNOS, which is the nitric oxide, superoxide, producing the peroxynitrite. And this peroxynitrite is a very fast, short-lived, reactive, produce the mitochondrial effects, which produce the depletion of ATP. It can nitrate the proteins, it can peroxidize the lipids, and it can produce the energy, by reducing the ATP. So to look at this, we have two different approaches. We do a pharmacological approach. We have a compound called 7-nitroindazoles. We block the NOS and see if it produces any neuroprotective effects. And then we also were able to genetically form these NOS knockout mice and see if they have any effects on the methamphetamines. In case of 7-nitroindazole, as you can see, you get dopamine. 7-nitroindazole has no effects. METH produces nice effects, and if you put together, it should be a protective effect. So definitely, if you can block the nNOS, you can see the neuroprotective effects. Same thing with dopamine transporters, control 7-ni, METH as well as it. In case of transgenic mice, we have the same effects. We have these are the transgenic, homozygous, and heterozygous, and these are the three different mice we inject the METH because it takes 3 different mice to produce this type of mice. You can see a significant decrease in dopamine, while in case of heterozygous partially and homozygous completely protected. So definitely NOS is playing a major role in nitric oxide. And you can see the same thing with dopamine transporters. You can see the C57, SP129s, and NOS has no effects on dopamine transporters. So it's always, like science has, every time you prove something, you establish something, so what is next? What is next? So what was the next? I said, well, how really, we proved that nitric oxide is responsible, peroxynitrites may be forming this, what is really happening, how it affects the dopamine system. As I showed you before, you are producing the peroxynitrites, and this may be responsible. So proof that points, we perform several, actually first the chemical experiment, just to understand how it is working. You get peroxynitrite, superoxide, nitric oxide, you're producing the peroxynitrite. And we thought this is possible that this might be affecting the tyrosine hydroxylase, because this is the enzyme which is responsible for synthesizing the dopamine. If you can nitrate the tyrosine, you can form a compound called 3-nitrotyrosine, which is still in the brain. So we tried to establish an HPLC system where we can measure the 3-nitrotyrosine in the brains. And we look at this, and we look at in the cell culture as well as in vivo. First we took the PC12 cells, give the methamphetamine different doses. You can see 10, 20, 50, 100, and you can see a nice decrease in dopamine. And the same set of cells, when we look at the 3-nitrotyrosine, you can see a nice increase in 3-nitrotyrosine. So it means that you are depleting the dopamine, nitrating the tyrosine, and increasing the 3-nitrotyrosine. So we did series of different antioxidant compounds. Because of the time, I don't want to go each, but I'll just give you an example with the melatonin, selenium, 7-nitroindazole, and some of the compounds, the iron compounds, which also block the 3-nitric oxide formations. We also published several papers on astaxanthin. It's a very powerful antioxidant, as well as N-carnitines, which showing that it definitely can block the nitric oxide, peroxynitrites, and block the effects of METH on dopamine system. Here is the melatonin experiments, where we look at the control melatonin. Again, after methamphetamine, you can see a nice decrease, but when we combine together, it shows protection. So this is the dopamine concentration, and when we look at the 3-nitrotyrosine, you can see, okay. So it's a really good correlation. You see the depletion of dopamine, you can see the increase in tyrosine hydroxylase, the 3-nitrotyrosine. We look at the selenium again, we look at the control methamphetamine and increase the concentration of selenium. You can see the dopamine is showing the protections, and when we look at 3-nitrotyrosine, you can see the same thing. So you can see that definitely if we can block the nitric oxide, peroxynitrites, you can block the effects of methamphetamine on dopamine system. We also look at the selenium in mouse in vivo experiments. We took the control animals, selenium, meth. You can see a nice depletion, while in case of combination, it showed you protective effect.
Excuse me, what is its effect on monoamine oxidase?
You mean the methamphetamine?
Yeah.
It decreases it decreases the activity of it. Actually, there's also an effect of 7-ni on monoamine oxidase. We published that actually. Again, it's showing the nice increase in 3-nitrotyrosine. So really, we were using these two biomarkers together, combined, showing that dopamine depletion and 3-nitrotyrosine increase to prove the point that 3-nitrotyrosine is important as a biomarker for the neurotoxicity of the METH-induced dopamine toxicity. Again, we use two different types of transgenic animals. We start with C57, NNAS, SOD overexpressed. These are the controls. This is METH, and this is together. This is a very tight result that was surprising. This is the dopamine data, and this is 3-nitrotyrosine. So you can see that we are over and over and over with series of papers showing that definitely if you affect the dopamine system, but if you look antioxidant system, which can affect the nitric oxide system, you can block that effect. So once we established that, with the help of a stereochemist at UAMS, they have a crystallography. So we said, well, let's see what is doing to the tyrosine hydroxylase, because when we measure the tyrosine hydroxylase, we can see the decrease in activity, but Western blot showing no effects. No total activity the same. So we say, well, how we solve this problem? So we look at the structure. This is the tyrosine hydroxylase, and these are the nitrotyrosine. If you look at a little bit higher magnification, again, this is the active side of tyrosine hydroxylase, and these are the tyrosine on the tyrosine hydroxylase. And when we look at the data in after control and experimental, based on the stereology, or they use the crystallography, they measure the distance of the tyrosine from the active site. And because of that, it was dismantling the activity of tyrosine hydroxylase. And it was very interesting because this is what we published this paper, and within a week or two, there was a paper came from Michigan showing the same result using a different techniques. It was so nice to see that there's somebody else showing the same thing what we have shown. So what we proved that what is happening, the methamphetamine increasing the nitric oxide, crossing the peroxynitrite, is that peroxynitrite dismantling the tyrosine hydroxylase activity because changing the distance from the active site to the tyrosine. And that's what we are forming the 3-nitrotyrosine. After this, we look at all the gene expressions. We have looked up almost like 20,000 genes, but I just want to give example which is very important. P53. P53 is a tumor suppression protein which induces the cell cycle arrest and apoptosis when overexpressed. And METH has been reported to regulate the upregulate the P53 in striatums after METH administrations. And here is the results. You can see this is control, this is up. You can see METH is upregulating the P53, where in case of transgenic mice, we see no effects. We look at the another gene which is more downregulated gene. BCL2 is a B cell leukemia oncogene which block the apoptosis, and it decreased or detoxify the ROS, and repeated doses of METH can cause downregulation of the dopamine in the stratums, and BCL2 has antioxidant effects and protects against the oxidative stress-induced cell injury, and here's the results. You can show this one. So really it was a good correlation between BCL2 and P53. So this is the bottom line really. What we found that when you have the METH to the dopaminergic systems, it flood the dopamine, and because of that, it has a massive release, produce the auto-oxidation. This auto-oxidation can start the generation of free radicals, which is the superoxide radicals, and you have an NO available, which produces the peroxynitrites. You can block these effects in a different way. You can block this NOS, so you can block this increase of NO, and you will have no peroxynitrites. We also have done some experiments in cytokinase are playing a major role because the calcium reflex, because that will control the activity of NO. So the bottom line is really that these in vitro as well as in vivo data clearly support the hypothesis that peroxynitrites play an important role in METH-induced dopaminergic toxicity. Selective antioxidants such as melatonin, seleniums can be useful neuroprotective agent against the METH-induced toxicity. Manipulation of NOS, neuronal NOS, such as using the pharmacological FTO7 can be a neuroprotective strategy for METH-induced dopaminergic toxicity. Some of the iron compounds can be used. And so we really thought 3-NT can be used as a biomarker for peroxynitrite generation and should be investigated in other neurological diseases. And when we published this paper, several people came out that it increased in Alzheimer's, it increased in Parkinson's disease, and all other neurological disease. So this is really a good biomarkers can be used. Yes, sir.
So how methamphetamine is stimulating, activating the NOS and NOS?
How is activated? No,
it's producing in nitric.
increasing the yeah.
Is it expression or activity or what is it?
It's total NO. It's generating the NO in the dopamine cell, in the cell actually. When you have a massive release of dopamine, you really producing the superoxide, and you have NO available.
Yeah, but then dopamine should be then activating something.
Yeah, well, it's generating the superoxide and hydroxyl radical, and that reacting to NO, which is already present in this in the system.
So
anyway, so, and I want to acknowledge all the people who really do all the work, so all the collaborators who are responsible for a lot of, mice as well as other work. And this is a beautiful picture from the coast of Chile. Alex may appreciate that. This is the coast of Chile, Santiago. And I want to thank you for your attention and when you put people to sleep. I will be happy to answer any question, and I will be happy to share you the dopamine song if you are interested.